All right, in this video, I'm going to explain the reactions that occur during glycolysis, or the breakdown of sugar, which takes place in the cytoplasm of all cells, plants, animals, fungi, bacteria, in both aerobic and anaerobic conditions. So it means that means it can take place in conditions where there is no oxygen present or where there is oxygen present. Glycolysis will occur. The purpose of glycolysis is to break down glucose into a smaller molecule called pyruvate, which is 3 carbons. So it basically takes the 6-carbon molecule glucose, breaks it in half into a 3-carbon molecule called pyruvate, and extracts some energy along the way. But importantly, now pyruvate is small enough to enter the mitochondria and then complete the oxidation of pyruvate in the Krebs cycle and oxidative phosphorylation. It begins with a glucose molecule, and I'm only going to show you the carbons and phosphate groups that are added here. So there's a glucose, an activation energy of 2 ATP is needed. So they're going to be shown like this. ATP on this side, it leaves as ADP, and the phosphate group will be donated to the glucose, and one will connect on the left, one will connect on the right, and it actually rearranges glucose into its isomer fructose to form fructose 1,6,5-phosphate. So the 1 and the 6 refer to the phosphate groups connected to the first and 6th carbon in this isomer of glucose called fructose. This is then split. There's an enzyme that breaks it in half, and we're left behind with 2 3-carbon molecules, each with a phosphate group. Should be familiar from the Calvin cycle. These are both p-gal molecules. So what we just did, we just invested 2 ATPs worth of energy, and by the end of this reaction, we're going to actually get out more than 2 ATP. So there's a net gain of energy that's coming out of this reaction, but 2 have to be spent to go in. After p-gals are formed, a phosphate group is added, but without the addition, or out the need of, I should say, without ATP. An enzyme will take a phosphate group and attach it to each p-gal, leaving behind BPG. And this is what we call the end of the energy investment stage. Just energy investment stage. Energy's been invested. Now it's time to get a return out, for the cell to get a return of energy. So that's the second phase of glycolysis, is energy payoff. So let's begin again with BPGs. Two of them. Both are going to happen simultaneously. The first electron carrier, called NAD+, will oxidize BPG, which means it's going to pull off 2 electrons and 1 hydrogen ion and form NADH. This will happen to both PPGs, so you can say this oxidation process happens twice. So we have 2 NADHs formed. This is no longer the same molecule. It's going to look the same because we still have 3 carbons and 2 phosphates. But it's given a different name because it's slightly different chemically. This is PEP, the one that's found in the C4 cycle in C4 plants and CAM plants. It's PEP, or PEP. The last thing to do here in this reaction is to grab and unphosphorylate these PEPs with P's. So 2 ADP will come in. Each one will collect one of these phosphate groups and leave as an ATP. Same thing will happen on this side. 2 ADP. P will connect with ADP and leave as ATP. And we finally have our product. 3-carbon molecule there, 3-carbon molecule there. Each is called pyruvate or pyruvic acid. Same thing. All right, so let's look at our products here. What actually happened in this reaction? The net products accounts for everything that went in, everything that comes out. So we say that we gained a net of 2 ATP because 2 were spent to get this reaction going. We actually formed a total of 4 ATP here, but at the very beginning stage, 2 were needed. So we say that 2 net were generated. 2 NADH were formed in the payoff stage here and here. And then 2 pyruvate are formed. All right. After the ATP are generated, the cell can use them to do whatever the cell needs to do. So they go right to cell work. The NADH has to be converted to ATP by oxidative phosphorylation, and we'll get to that in another video. And the 2 pyruvate, depending on the amount of oxygen available, if there's oxygen available, the 2 pyruvate will enter the mitochondrion, and the next step will be the Krebs cycle, or the citric acid cycle. If there's no oxygen available, these 2 pyruvate will enter a process called fermentation.